Hello, my name is Rebecca Arroyo. The main takeaway from my presentation is that cardenolides, which are defensive chemicals from cardenolide secreting plants, are sequestered more by some insects than in others. Sequestering means that they take in the toxins and use them as their own, which usually translates into them being unpalatable to their predators, specifically in these insects. The monarch butterfly, also known as Danaus plexippus, the queen butterfly, also known as Danaus gallipus, the common crow butterfly or Euoplia cor, or Radiolus superbus, and Ligeus equestris, the red and black bug. This is a figure that I created for my report. In the graph, it is evident that the black and red beetle has the most cardenolides sequestered, with Horvathiolus superbus following. The monarch butterfly is next, with the queen butterfly having the second to least cardenolides sequestered. The insect with the least, or in this case none, cardenolides sequestered is the common crow butterfly, also known as Euoplia cor. The amount of cardenolides sequestered by different insects matters because it shows how these insects adapting to plant defenses can affect the whole ecological system. It also shows how some insects do not adapt to sequester, just as seen with Euoplia cor. The constant evolution shows that these plants might become endangered because they cannot protect themselves anymore from herbivory. Competition could also negatively impact the environment because these insects help to pollinate the plants, which leads to cleaner air, healthier soil, and carbon sequestration. Competition can limit these benefits. From the first paper that I used for my report, I pulled the data from this figure. First, I used the data points given from the cardenolides found in the gut content. I then calculated the average for these three insects shown and ended with micrograms per milligrams of dry mass as my units. Finally, I calculated the standard error with standard deviation and sample size. On the bottom is where the citation for my paper can be seen. From my second paper that I used for my project, I pulled the data from the figure on the left. As you can see, there's a lot of data on this, so the only one I used is from graph C because that's the only one that uses cardenolides. I used the ones that were for L. equestris and H. superbus. Next, I calculated the average for both insects and ended with micrograms per milligrams of dry mass as my units. I then calculated the standard error with standard deviation and sample size as I did with my previous paper. And I ended with a NOVA test at the end for my raw data, which is where I got my p-value, which is lower than 0.05. And for my null hypothesis, it was rejected because not all the groups are equal, which can be seen with my p-value, which is lower than 0.05.